Thank you, Missy. All right, Steve, whenever you're ready. Hey, Bozeman. I'm Tanner Wiegand. As Missy said, I'm a scientist, and I study viruses, but not the kind of viruses like SARS-CoV-2 that was responsible for the recent pandemic. Instead, I study the viruses that infect bacteria. We call these phages or bacteriophages. They're found in about every environment on our planet. So bacteriophages come in a dazzling and haunting variety of shapes and sizes, but they're one of the most abundant biological entities on Earth. And in fact, there's an estimated 10 to the 31st phage particles at any given moment on our planet, which equates to roughly 1 trillion phages for every grain of sand. And these viruses have co-evolved with the bacteria they infect since the genesis of life on Earth. In response to that selective pressure, bacteria have evolved a number of immune responses that they use to fight off these viral infections. And we think of this like an arms race where one side innovates and the other side counter-innovates. But the immune system that my lab studies is called CRISPR. And CRISPR has been in the news a lot. Some of you may have heard of it due to its applications in genome engineering. But it was initially discovered in the context of the bacteria that are used to make yogurt, where it functions as an antiviral immune system. Before I tell you about what CRISPR is and how we've repurposed it, though, let's all get on the same page about what a virus is. These are metabolically inert parasites. They're pretty simple, usually just composed of a protein shell that encapsulates the genome. That's made out of RNA, or more usually in the case of bacteriophages, DNA. And when a virus attaches to a host cell, it injects its genome into that cell. The virus then begins expressing its genes, taking over the host cell resources to make more copies of the viral genome, as well as the proteins that make up that shell. When enough viruses have been made, it bursts forth and infects neighboring cells. So CRISPR systems are the only adaptive immune system we know of in bacteria, and they function primarily in their first stage called adaptation by stealing a snippet of the viral genome and integrating that within the bacterial genome. Now, these viral memories act as sort of vaccination cards. Those DNA memories get expressed into RNA molecules that then guide proteins like Cas9 to target viral DNA sequences that match the sequence of the RNA guide. By cleaving the viral DNA, they effectively shut down the viral infection. CRISPR gets its name from the way these memories of viral infection are organized in the bacterial genome, since each of these vaccination cards are interspaced by these short palindromic repeats, leading to this mouthful of an acronym. Why is CRISPR such a big deal? About a decade ago, we figured out that we could take these systems out of bacteria and port them into human cells, allowing us to cleave any sequence of DNA programmably that we wanted to. This immediately opened up the door to curing genetic diseases like sickle cell anemia. So the way these treatments work is that a patient comes in and has the stem cells from their bone marrow extracted. Those are edited outside of their body simply by swapping out this vaccination card or that guide RNA molecule of Cas9. We can now direct it to cleave genes that are associated with the disease. In the case of sickle cell anemia, we don't actually edit the gene that is responsible for red blood cell formation in adults. Rather, what we're doing is turning back on a red blood cell gene that's only active during fetal development normally. So these treatments have been used on a number of patients now, and in December of 2021, Victoria Gray became the first person in the world ever cured of sickle cell anemia. Due to the success in both her story as well as the stories of other patients, the FDA has now approved this cure for widespread usage. Scientists, including myself, are now turning our lenses on the 4,000 other monogenic diseases, or diseases that are caused by a single mutation in our genomes. And in recognition of their discovery and initial characterization of CRISPR, two scientists, Emmanuelle Charpentier and Jennifer Doudna, were awarded the 2020 Nobel Prize in Chemistry. So the utility of CRISPR really comes from the fact that every living organism on Earth encodes its genes in DNA, which means that beyond bacteria and humans, we can take these systems that evolved to manipulate DNA and port them into either crops or livestock or whatever organism you can imagine. And outside of the flavor of CRISPR that I've described tonight, Cas9, many other evolutionary trajectories have produced tools like Cas3, which can conceivably delete entire chromosomes, curing things like Down syndrome, or Cas systems, which can move large pieces of DNA, which could enable us to help crops resist climate change. So my CRISPR journey began in 2017 when I started my PhD here at MSU, and I'm fortunate today to work with an amazing group of colleagues at Columbia University, where we study the molecular mechanisms of these systems, trying to understand how they function as antiviral systems with the hope of better informing how we can repurpose them as biotechnologies. As Missy said, I'm primarily a computational biologist, so I don't do a lot of pipetting these days. Instead, I'm looking through genetic code to discover new flavors of CRISPR and leveraging advances in machine learning to understand at the atomic level how these things work. We face a few challenges still in the CRISPR field. One big one is delivery. How do we keep Cas9 from targeting cells that we don't want them to? So your liver, for example, if we want to target that, how do we keep it from going to your brain or heart? Cas9 also cuts pieces of DNA we don't want it to. But I'll just close by saying that nature has created an incredible bounty of nanomolecular machines that we're just beginning to understand. All of that research is dependent, however, on continued funding of science education and research. So as we approach a contentious election, I encourage you all to vote for candidates that are responsible stewards of that natural bounty.